So good morning, everyone, and good afternoon to everyone joining us today from around the world. My name is Jessica, and on behalf of Gasako Research, I'm delighted to welcome you to today's webinar on the development of specialised tools and methods to advance your understanding of the canine gut microbiome. I'm joined today by Shema Ahmed, Product Manager at Diversigen, Jan Suchodolski, Professor and Associate Director of the Gastrointestinal Laboratory at Texas A&M University, and Emily Hollister, who is Director R&D Computational Biology at Diversigen. After the webinar, we'll have a short Q&A session. Please do submit your questions throughout the webinar, and we will address them once the presentations have concluded. We do also have polls included throughout, and I'd encourage you to please fill these out when they appear on the screen. It's great to have your involvement throughout the session. The full recording of the webinar will be made available to watch on demand once the webinar has concluded. And now, with no further delays, I'd like to hand over to our speakers to begin the presentations. So Shema, Jan, Emily, over to you.
Perfect. Thanks, Jessica. So my name is Shema Ahmed, and I'm a product manager at Diversigen. So for the webinar, we'll be having our team of experts, Jan Suchodolski from Texas A&M and Emily Hollister of Diversigen, who'll be walking you through the importance of studying the canine microbiome and what are some current limitations and challenges to us truly understanding the canine microbiome, and how Diversigen has addressed some of these challenges through MetaGene Canine, which is a tailored metagenomic service. And then we'll finish off by discussing a case study that really emphasizes the importance of studying the canine microbiome. So before we delve into the science, I just want to go over who is Diversigen. So Diversigen is a microbiome service company that offers many different services across many areas of the microbiome to help push microbiome science forward. And so for today, we'll be focusing on our ability to help push the canine microbiome science forward. And at Diversigen, we're made up of many different experts across many different areas of microbiome research. So without further ado, I'll pass it on to Jan, who will be discussing the importance of studying the canine microbiome.
Thanks so much. So welcome, everybody. Yeah, so I do microbiome research now probably for 20 years, and I think it's a nice story where we started. We started even in my first year of PhD with bacterial culture because we thought it's so simple. Then we moved on to sequencing. Then we moved, for many years in the metabolomics and so on. And I think we made a lot of progress, and I think we have some really good tools already to assess where are we in the microbiome. But of course, it always depends, like in any other organ system. At the moment, I think we are know where is the huge range of normal. We know for sure what is abnormal. But again, to improve health and so on, sometimes we need to afterwards also look on the finer details. So where are those, niches that we cannot yet access with those tools, and how can we improve them? And why should we do that? Because we know the gut microbiome is really important. I think in the past, we looked mostly at pathogens. Now we're discovering that it's really an organ system. And I think that's really something that we need to highlight, because often still we think it's an independent system. It's out there. It provides a few benefits. But I think the most important thing is that it's really part of the organism. We know that if you have a good microbiome, it provides growth. It's a metabolic organ. So there's a lot of metabolites. I think those are probably the most important one to look at. And through those metabolites, it can definitely influence immune development and function, and not only in the GI tract, but ultimately also outside in other organ systems. But I think what is really important to understand, the microbiome is not isolated. I think the biggest challenge, in my opinion, is, from my experiences, is the focus only at the microbiome itself. Okay, and I think it's really challenging because the microbiome is not isolated. It is part of the whole organism. And so what that means is, yes, it's important for our health being, but it's also important to understand what happens inside the gut also influences the gut microbiome. I think often we talk about nutrition, diets, antibiotics as the major drivers, but actually the more we understand it, I think the first most important is what is the underlying gut environment, the gut structure and so on, because that really influences the gut microbiome a lot. And my experience is actually it's not easy to change the gut microbiome. And I'm going to talk to this a little bit later, but it's important that we understand that we look at the gut microbiome, because in a healthy state, a lumen of the GI looks like this. We have this very diverse gut microbiome with all these functions. But we also have this very important mucus layer here. And this mucus layer is probably one of the most underappreciated parts of the gastric barrier, because this mucus layer really separates all those bacteria and all those metabolites that are present inside the lumen from the epithelium. So we need this mucus layer to really have this separation. Then we have, obviously, importance of permeability. We need a tight barrier, because that really prevents translocation of dietary or bacterial antigens, and ultimately we have a well-regulated immune system. Now the challenge is, I think, if the gut environment is healthy, it's actually quite easy, or not easy, but we can modulate the gut microbiome to some degree. So, but if there's GI disease present, and in this case I'm talking about chronic GI disease, chronic enteropathy, like we call it in dogs, it's not just a dysbiosis, it's not just changing the gut microbiome. There's this degradation, this mucus layer in many instances. We have in some, depends on the severity of the disease, increased permeability, and all of this ultimately drives change in the immune system. And I think it's very important that we have tools to differentiate. Is the gut environment normal? Then yes, we can to some degree modulate the gut microbiome. Is the gut environment very abnormal? Then often this is a major driver of bacterial changes. And yes, the microbiome can help us restoring health and so on, but it's not going to be sufficient to just modulate the gut microbiome. And we see this with many studies, like FMT, for example, that really it's very difficult actually to bring it back in GI disease. So again, what is important is to have markers to understand all the pathophysiology that happen at the same time. And that means we need to understand who is there, understand the function of those bacteria, and ultimately we need also markers that are assessing the gut integrity, like inflammatory markers and permeability and so on. And this is a really good example that how we have to look at, what is the state of the dog in general. In a general healthy dog, you see this this is a biopsy from the colon of a dog. You can see here in blue is the epithelium, and then in red are the bacteria. And you see those bacteria are really far away from the epithelium, because this is this important mucus layer here. And this mucus layer really has this functional barrier away from bacteria. And so in this case, again, it's a general healthy gut environment. I think it's quite easy with nutrition, diet, change microbiota function, and so on. But imagine you have a dog with chronic GI disease, and that's actually also a true case. You can see here this epithelium, and as you can see, there's bacteria in red, and there's really no separation whatsoever between the bacteria and the gut environment. So again, very different conditions. We need really sensitive tools because it's difficult to assess the inside of the GI tract. So ideally, we have to have a multimodal approach that really encompasses different markers, bacterial gene markers, and also host markers to truly able to assess what is actually going on inside the lumen of the GI tract. So over the time, obviously, when we started with culture, we started with 16S sequencing. 16S sequencing is really a great tool, I believe. It helped us a lot. And so across many studies, if we did studies repeatedly, we are able to identify some of those genera, some of those major taxa that are usually go up in disease or go down in disease. And we are able to identify some of those. However, one challenge with 16S sequencing, it's a really quick procedure. It's nice. I think now it's very, very very kind of cheap and so on. But we still have this little bit of limitation that we do not really know what is going on down on a species or strain level and so on. So that's why we started over the last few years, together with Diversigen, also do much more shotgun sequencing. So DNA shotgun sequencing has the advantage it has no PCR bias. It really gives us the species level identification. It used to be super expensive a few years ago, but I think now with the technique of shallow shotgun sequencing, it's really nicely accessible. And in the end, what happened to us is nicely because it confirmed to some degree what we saw in the data of 16S sequencing is that we can actually identify so-called core microbiome bacteria. And what that means is we know the microbiome is very diverse. There's like several thousand species that we identified across 12 healthy dogs. But if you look very carefully, and we saw this already in 16S sequencing, but also shotgun sequencing confirms it nicely, those in green are present, those in red are absent. And when you look very carefully, there's really not that many species of bacteria that are really consistently present in every single dog. So in the end, we identified roughly 25 of bacterial species that are really present in all 12 healthy dogs. 36 of them were present in 9, 12 dogs. But that's kind of probably the major bacterial, the core microbiome and so on. And then we identified some of those bacteria that we already kind of had a hint in the 16S sequencing data and so on, but those are really the bacteria that are really top of those. So some of them I'm going to discuss a little bit later, Clostridium hydrononis, Blaut, and Faecalibacterium. Those are the top abundant bacteria on a species level inside a healthy gut. And so parallel to that, we actually, over time, we had actually qPCRs developed for some of those bacteria. Because I think one important strategy is also that we have to have a discovery tool. That means a broad discovery tool that can help us to detect unknown things. But also, in order really to bring the microbiome into the clinic, we have to also have reproducible tools where we can look at individual dogs over time. That means I want to have an assay that I can measure today, and I can measure in 6 months or in 2 years, and it still gives me a good reproducibility. And that's why we actually, over time, we looked at qPCRs, so quantitative PCRs. And again, those quantitative PCRs target some of those core bacteria. So some of them, like Fecalibacterium, Clostridium is really one of the most important bacterial groups in a dog. They usually go down in disease or in dysbiosis, and some of them are more harmful, not necessarily per se that they are really pathogens, but they're usually an overgrowth bacteria when there is some dysbiosis pattern and so on. And what that allowed us to do is not just only have reference intervals for some of those groups that are really validated across many different countries, but also we can put these numbers together. So we can do so-called dysbiosis index, which in the end is each dot here is the combination of all the results of all those bacteria together. And so there's a mathematical model. And we can, for the first time, to some degree, define where the huge range of normal is. As you can see, and we validate this in many different countries. So this is actually very fascinating. It really works really well across the world. And most likely because most dogs and cats, if they're commercial diets, they're quite similar in macronutrient contents and so on. And with the dysbiosis index, we can also see how much the gut microbiota shifts away from normal. So an increased dysbiosis index indicates that the bacteria shifts from normal. And this correlates very nicely with sequencing and also diversity. So the more the index is increased, the less diversity in the microbiota based on DNA shotgun sequencing or 6NS sequencing. And so this is one of the first steps, because I think it's important, like with every other organ systems, we need to understand is where is the huge range of normal? When can we very specifically say it's abnormal? We know that above 2 is highly specific. That's an abnormal microbiome. But as you can see, there's many different disease phenotypes that we associated with microbiome changes, but they might be more subtle. And so, for example, epilepsy, there's this gut-brain axis. As you can see, all of those dogs are within the normal range on the dysbiosis index, so like in healthy dogs. But there are some some initial data would suggest there is some changes in the short-chain fatty acid content, bacterial function, and so on. So again, what is needed as a next step, in addition to knowing where the normal is, but also if those that are normal on the microbiome shifts, are there some fine signatures that we need to yet identify? And that is really, it's going to be important to add gene function. So looking at bacterial gene functions and their functions and so on, and into the localization. So with this, I'm handing over to Shaima for the first poll question.
Thanks, Jan, for that great introduction into why it's important to study the canine microbiome. So our first question is, what microbiome sample type are you interested in analyzing? Is it stool, or the oral microbiome, skin, or other? And if it's other, please let us know in the group chat what your other is. So I'll just give 10 seconds for you to answer that question. Great. So it looks like the majority of people on the webinar are studying stool, and second is skin, and it looks like tied for third place is oral and other. Great. So now I will pass it on to Emily to discuss some of the advancements and challenges in studying the canine microbiome.
Thanks, Shaima. Hi, everyone. My name is Emily Hollister. I'm so glad to join you today and share what we've been working on in terms of trying to advance the field. Not us, but others certainly have contributed to incredible discoveries in the microbiome field and across a variety of hosts and environments, powered by a number of different sequencing platforms, including Amplicon and shotgun sequencing, as Jan mentioned, but also metatranscriptomics as an emerging and re-emerging tool. Certainly, bioinformatics tools and analytics tools are key to this. Once you have all of that data, how do you interpret it, and how do you find those signatures that are perhaps most important or indicative, or providing those next key mechanistic steps as you try to understand the systems level biology of your system or disease of interest. And of course, powered by culture studies, cohort studies, culture techniques, and model systems. We know that germ-free rodents can be really important in terms of translation. And ultimately, the whole reason we're here today is to talk about MetaGene Canine, because we know that while generating all of this data is great, if you're only able to use a partial lens on your community, you may be missing out on really valuable information. And so this was a big motivator for a number of reasons. A curve like this is probably familiar to most people in the audience, but when we look at microbiome publications across species, and this is a very informal poll of PubMed with keywords, human microbiome in the title or in the abstract, what we see is this very large rise from, starting in 2010, although microbiome publications did exist before that, but, into the tens of thousands or hundreds of thousands, rather tens of thousands of microbiome publications per year annually at this point. If we look at mouse studies, in contrast, there are far fewer, although really important advances have been found in model systems. And if we look at canine, the curve looks relatively flat, although that is deceiving. So if we zoom in on this and use a log scale, we see that there is a steady increase in all of these spaces and roughly at a similar pace. So we know there's interest and we know there's desire, but ultimately the big question is, can we advance our understanding? Can we shine more light into the space and enhance our ability to capture information from systems like mouse or canine, those non-human systems that for organisms that we care so deeply about? So a quick additional poll question before we talk about the MetaGene Canine solution and the new database that we've built is, what do you think are key limitations to microbiome studies today? Is it that databases are not representative enough? Is cost an impediment? Is it the logistic aspect of collecting samples from large cohorts? Is it a funding situation, lack of reproducibility, or certainly recognize that there could be multiple, but if you could pick the one that you think is most key to the largest barrier, we'd appreciate that feedback. I'll give you just a few more seconds. I'm actually very curious to know. And let's click through. Reference databases. Awesome. Well, it's not awesome in the fact that there is that gap, but hopefully what we'll be sharing today is really exciting for everyone here. Certainly reproducibility and logistics are other challenges. And hopefully with a variety of movements in the field around better data capture, standardizing approaches could help. We may be able to tackle some of that reproducibility question. So as we've highlighted and alluded to, we know that there are challenges in canine microbiome research. And one of the key ones from my perspective, because bioinformatics is key to my heart and the things I think about on a daily basis, are the representation of our databases. Really, truly, up until now, there's been essentially one canine relevant gene catalog that was developed. This was developed a few years ago, and with a really great solid study out of Europe where they looked at 64 dogs and approximately double that number of samples to create a gene catalog. The interesting thing about it is that canine and human gene catalogs share some content. And so what these authors found was that when they mapped canine samples against a human gene catalog or a human reference, they did okay at information capture, getting somewhere in that 65% of canine reads mapping to this human catalog, which is good, but that begs the question of what's in that other 35%. And we see, there are quite a number of host-specific genes or genomes that we know are not captured. So this was the big motivating factor for us to develop the MetaGene Canine solution. And it is an end-to-end solution where we can handle, extraction of canine gut samples in a high throughput manner if needed, extracting consistent high quality DNA libraries, working through our sequencing and annotation pipeline to ideally develop or generate insights. And key to that is this MetaGene Canine database that we've developed. And to tell you a little bit about how we did that, we started with a base of RefSeq, where we curated well-known microbes, broad coverage. This applies to any number of systems. We then built on top of that, pulling key bacterial taxa that have been identified in the literature, either from culture-based studies or inferred from key taxa identified in 16S, finding those relevant genomes to go along with it. And then on top of that, we added in MAGs, so metagenome assembled genomes. And what we've found from a variety of studies across time and space and host is that MAGs can be really key to improving your annotation rates and be moving beyond many of the culture-based limitations. Those who are familiar with this field know that culture is fabulous for getting a single isolate, something that can be sequenced, but it may not be the bug of interest. And many of our isolate collections are very biased toward pathogens. Of course, it makes sense if you're trying to get rid of the thing that is making you or your dog or your plant sick. You want to get rid of it and really understand it. But ultimately, as Jan mentioned, we're recognizing that the microbiome, and especially the gut microbiome, but certainly other body sites, is really kind of an additional organ and a body system that's worth understanding and fostering and facilitating. So we need to understand the breadth and depth of what and who are there to really ultimately be able to improve health and outcomes. So for those who are less familiar with the idea of MAGs, the data I'm showing here is based on samples derived from human subjects. But what we find is that there's tremendous diversity and tremendous untapped diversity in MAGs. What you see on the left-hand side is a figure from this Nature Biotech paper published last year, where the authors found that a large proportion of novel content can be found in MAGs versus what is known relative to genome isolates or bacterial isolates. And when we map samples collected from around the world to a reference genome collection that included MAGs, what they found was up to 400% improvement over a standard RefSeq annotation. That's huge. Also bringing mapping rates into the 70 to 80+% range, which is also fantastic. Quick note about MAGs, though, is that they can vary tremendously. And this is an overview of the definitions of MAGs. It is a challenge, and I like to use the analogy of a jigsaw puzzle, right? When we sequence a single genome, the challenge is to put all of those reads back together to create a complete picture of the microbe we're interested in. When we do this from metagenomes, this is like taking 1,000 jigsaw puzzles, or maybe even a few hundred, putting all the pieces in one pot, tossing out about half of them, and then trying to recreate the pictures. Can we get a full view and a full representation of the individual puzzles or genomes that we started from or that we're trying to identify? In some cases we can, and high quality or near completely finished genomes have been assembled back out of metagenomes to date. So these definitions range from low quality to finished. And I will note that high quality is a very high bar to meet from the perspective that it requires ribosomal RNAs as well as tRNAs, and a large number of them to hit this threshold. However, there are a number of very good medium quality MAGs that exist in the literature and through various data sources and through various assembly efforts. I'd note that we focused on the medium to high quality MAGs in our build with our database, including MAGs that have a median completeness of 93% and an average contamination well below 5%. So really the whole goal of this is to get to a point of enhanced information capture. So to give you a sense of where human samples fall on this information capture spectrum, I shared the data from that 2021 Nature Biotech publication. And to zoom in on a more isolated or more specific instance of that, if we map human reads to a curated RefSeq database, we get read mapping rates in the 50 to 60+% range on average. And really the goal is, can we achieve the same type or better level of annotation with canine gut samples? And the answer is yes. So if we do the same comparison, mapping canine samples to that curated RefSeq database, we get annotation rates hovering around 30% on average. But with our MAG and key genome augmented solution, we're finding annotation rates at 60+%, which is well in the range of what we see for human. Of course, it's not a full solution, but it's giving you far more information than what we would see otherwise. And so this is for taxonomy with a focus on genus, genera and species. And if we look at function, it's a similar story where we're capturing tremendously more information into KEGG pathways. And a brief note I would highlight is that anytime you map against KEGG pathways, it's not common to see super high annotation rates because of the way the KEGG database is annotated and focused. Not every gene is linked explicitly to a KEGG pathway. A few more things I would highlight is that with this increased resolution and increased mapping rates, what we're finding is that we have better resolution to identify differentially abundant features. We do, in some cases, capture additional content, so in terms of KEGG modules, but what you'll notice in this sort of second column under differentially abundant is that the MetaGene Canine annotation captures many, many more features and much more gene information that would then potentially lead to deeper insight into your system because of the enhanced information capture. And so to put this into practice, it's not simply just the limited data that we've worked with internally, but this also applies to a variety of different use cases. So looking at healthy versus diabetic dogs, we see that same increase in information capture. Similarly, looking at time series following fecal microbiome transplantation, again, tremendous increases in information capture. And then finally, a time series around dietary intervention, which is a use case that a lot of people are interested in. And same story, regardless of the use case, what we find is that when we annotate canine fecal samples with this solution, we get a lot more information back out of it. So with that, another poll question that we'd love to have your input on is, how do you plan to use microbiome sequencing in your research? Are you interested in evaluating the impact of dietary intervention or modifications? Are you interested in the impact of medications on the microbiome? Are you interested in more basic biology questions, or are you digging at disease etiology, or perhaps something different? We'll give another sort of 5 to 10 seconds and then move into some additional content. Okay, dietary interventions, by and large, very interesting, and then very needed. I think we just don't know enough yet in this space. So with that, I would love to hand it back over to Jan to give you another example of how and why metagenomics information can power insights. Jan, take it away.
Thank you, Emily. It was great. And I think really it highlights that there's a lot of progress to be made, but I think with those tools, we really can get deeper in this information. And I'll give you an example. So again, my lab is coming more for the disease side. So we're trying to really understand what really causes disease, how those changes are. And ultimately, we are a multimodal approach. We identified some key pathways. One of them that we focus in currently is the bile acid pathway, which seems to really play a major role for maintaining a proper gut structure. The other one is obviously short-chain fatty acid, which everybody knows. And I think a third major pathway that has been shown in humans, in mouse models, important, but also really seems to be an important one in dogs and also cats, is the whole tryptophan pathway. And because tryptophan is a really fascinating amino acid, it usually comes from the diets, and it can be metabolized either by the host or by bacteria in different pathways. So it plays really a central role. So, for example, tryptophan can be modulated into kynurenine, which ultimately often is increased in IBD. It can be transferred to serotonin, which we know is a very important hormone pretty much for gut motility, that regulates GI secretions, and also plays a role in the gut-brain axis. So again, this is really dependent on tryptophan. And ultimately also, here where really is a beautiful example how dietary components that are pretty much utilized by the host, but also by bacteria. And again, that's why it's important to look at all of those three things together, because bacteria can convert tryptophan into so-called indole compounds. And again, depending on the levels, it could be ultimately very important for health benefits like mucosal barrier functions, for the tight junctions, for the barrier function, immune regulation, and so on. But again, it could be also, if the level is wrong, too much indole potentially could also be toxic and so on. So again, the tryptophan pathway is a really crucial one because it's a dietary protein that is converted by host enzymes or by bacteria in different ones. And the balance of all of this, because depending on where it shifts, okay, can really provide a beautiful benefit. And so one example is that, again, in the past, we looked mostly at taxonomy. So we identified some taxa that go down and up in disease. But again, after a while, it was not really sufficient anymore. We needed to understand more of the functions. We know that some bacteria can produce some of those bacteria. We know that also indole, depending on the disease phenotype, in healthy, in a fecal sample, should be actually quite low, because most of them is pretty much in a balance. Sometimes we have, in some disease phenotypes, we have increased amounts of indoles that we can measure using metabolomics, sometimes untargeted, sometimes we now have a targeted assay for that. And then you go to pieces in literature and other studies. The problem with, at least the challenge was always until now, that everybody focuses on one part of the system. So for example, there are publications out there that have nicely shown that when you measure it in serum, that serum tryptophan is decreased in GI disease. So dogs with severe chronic enteropathy, proteinosic enteropathy, have definitely a decrease in serum tryptophan inside the guts. But when you do the study only in serum, it's difficult to judge what's happening. Is there utilization by bacteria for more tryptophan? Is it because of dysbiosis? Is there actually loss because there's malabsorption of diseases? So again, looking at one side of the equation makes it very difficult. When we look at fecal samples, in opposite, we actually see that in some disease phenotypes, very acute enteritis, but also chronic GI disease, so long-standing GI disease, we can measure increased tryptophan in feces. So the combination of both would highly suggest that there is actually, probably, if there's low in serum, increase in feces, there's most likely some malabsorption happening. But again, we still need to understand the contribution of gut bacteria, because ultimately they can also utilize tryptophan. So we also need to look at the functional gene content of those bacteria. And so with metagenomics, so with DNA shotgun sequencing, we're actually able to measure this and say, actually, we can see that bacterial genes for tryptophan synthesis are decreased in dogs with GI disease. Okay? So again, we have a low serum level, increased tryptophan level, and a decreased bacterial genome for all those tryptophan. So again, it really helps us to create hypothesis and create a pathway, potential mechanisms, what is actually going on, where the tryptophan is lost. And so I think when we have a decrease in serum of tryptophan in GI disease, but an increase in feces, the highly suggestive would be there's a malabsorption. So our step is now confirming this with looking at the mucosal level and see is there decrease in transporters and so on. But it ultimately also tells us that ultimately, if we have a decreased absorption in the host, there's actually a lot of tryptophan floating around in the lumen, and bacteria kind of shift either taxonomy or decrease the shift in the utilization of tryptophan because there's so much out there. So my point of that is, again, looking at different tools, looking at metabolomics to measure direct in serum and in feces, looking at bacterial taxa, who is there, but more importantly, using metagenomics, so we can also identify what actually happens on a functional level in those bacteria. That's why this is such a powerful tool that can really help us afterwards really much better address and answer some of those pathways, and really afterwards be able to modulate it. So with this, that was my last slide. I think Shaima's gonna do the poll question.
Awesome. Thanks, Jan. Yeah, so Jan's translational study really focused on metagenomics paired with measuring different ways in which you can extract that information. So what we want to know is if you were to conduct a sequencing project today, which method would capture your goals? Is it shotgun metagenomic sequencing because you want to understand composition and function? Is it amplicon sequencing because you're working with a high host sample type? Are you interested in long reads because you want complete genomes, short reads for draft genome assemblies, or metatranscriptomics because you want to know what's being expressed and what's the activity of the microbiome? So I'll give a few seconds for you guys to answer that. Okay. So it looks like the majority of people on the webinar are really interested in metagenomics because you want to understand composition and function, but also metatranscriptomics, you want to know what's exactly happening at the gene level and gene activity. Great. So that brings us to the end of the webinar. So based on what Emily and Jan have really presented today, our solution really is that sample to insight, and with our curated canine database, you're really able to get that enhanced information capture to truly understand what's happening at both the compositional as well as functional level. And so I open it up for questions from the audience, anything that they're interested in knowing. And so we just have one first question. So I think this one, maybe Emily, you can provide some insight into is, I'm studying cats. Does your database that you developed, that you presented today for dogs, will it provide me with any additional insight into the cat microbiome?
It's a great question, and I would say that it's an underserved area. But what we have found from some internal testing and development is that it does help. So much like we demonstrated with the canine samples, when you use a sort of, I don't want to say generic, but a generalized reference database, you capture some aspects of your feline microbiome. I'm tripping over my words, but you don't get a full picture. Mapping against this database does help, and we are currently in the process of evaluating options to extend our database or databases to include a feline option with more targeted, with feline targeted content.
Awesome. Thanks, Emily. So we have a question from Elizabeth, and she's just asking, will the presentation be available in PDF? So I think the presentation, all of it will be available on demand, and that you can access all the content that way. So our next question is from Keith, and he wants to know, what do we know about breed-specific influences in the gut microbiome profiles? So maybe, Jan, you can take that question.
Sorry,
Jan,
I'm still here.
Yeah, no
Sorry, I need to find my unmute button under all the windows. So, great question. I think it depends really, like with every system. I mean, are there breed differences? Maybe. Are they very pronounced? So far, we have not found any. I mean, again, it depends like any other ecosystem or organ system. It depends on the tools we're using. And so when we look, for example, at major bacterial taxa, we never found any breed difference. Is it possible that because of functionality in the GI tract, there are some metabolic or some genetic functions? It's possible, if that makes sense. So again, it depends. In the big picture, we have not found any clear-cut breed-specific differences so far.
I would also add that there's a little bit of a challenge in that often many of the studies that have been published do have a very breed-specific focus. So as you can imagine, right, trying to control for as many variables as possible, many investigators will focus on their particular breed of interest or whatever their collaboration happens to give them access to. That's great in some regards, but it's also challenging in that, you know, the study I referenced, for example, was, I believe, all one breed. So we may be hindering ourselves in that sense, in that when we try to capture the broad diversity, we don't necessarily have the ability because this is still such an emerging field and the data sets are still growing.
Yeah, maybe I want to add to this a little bit because this is something that is really also a challenge. I think in veterinary medicine, we have small studies, sample numbers, and then each paper, compares one to the other, finds differences, but the size effect, how big is the size effect, is really missing. And it's really, I think, sometimes we have to be careful in the microbiome field when we see differences, they may not be really relevant. And so I think for the first time, what we have now with the dysbiosis index, we at least can put a number on it, how big is the shift. So that's like with any other marker. If you know what the reference interval is, you can much better judge. And I think what we need is really, also on a sequencing level, a huge study with many different breeds, and then we can make a conclusion if there are differences. But just comparing small differences, one breed to another, can be very misleading.
Great. So we have another question from Adam. And he's asking, after the insights are provided, what are follow-on steps for getting to actionable solutions to improve outcomes for dog owners? So Emily, I'll pass that on to you, and Jan, you can add after Emily.
Yeah, absolutely. And it's a great question and definitely a challenge. So my view on what we get out of sequencing today is that in many cases it's a discovery tool. So once we find those key taxa or functional genes or markers, there are a number of options for next steps. It may be that it helps develop a diagnostic. It may be that it becomes prognostic and can help us understand where your pet's health may be heading. From there, though, there's still, once we get out of that, or once we complete that discovery phase, there's a lot of work that goes into translation. I see Jan nodding his head. So I'll let him kind of pick up there. But I will say, I think there are a lot of opportunities, a little bit context dependent. Some will be faster to get into than others, especially if we talk about, say, diet intervention, for example. But if we're looking at, say, health and disease and moving into that space, it'll likely be a longer road to development. Jan, do you want to add to that?
Yeah, no, I mean, it's like with everything. I think we have some markers now that we know to some degree what they mean. And for example, I'll give an example. When you have a dog that has diarrhea and you measure Clostridium hydrononis, which is a really important bile acid converter, and by itself is probably, accounts for it. It's never changed in acute diarrhea. It's only changed in chronic diarrhea. Okay, and the most likely explanation for that is because you have some mucosal damage there, and that kills this really important anaerobic bug and so on. So I think we can use the microbiome to help us assessing what is the condition of the GI tract. So I think we are there. The future is going to be, for example, to say, if I don't have bile acid conversion, okay, I can maybe replace this bacterium because it's a key bacterium. At the moment, we can replace it beautifully with FMT. But again, the goal would be to say, I want to actually reduce this bacterium because I need bile acid conversion, okay? And then we could go in the future with designer probiotics, for example. We can say, maybe don't need this bacterium. We can actually just put probiotics that have this bile acid conversion capability and so on. So I think that is the future. I think we're starting. I think we have a lot of markers already. But again, it's like with every other organ system, it takes time to afterwards use those clinical trials to confirm that this is really going to make a difference.
Awesome. So we have another question for you, Jan. What other metabolite differences of tryptophan do you see?
Well, I mean, I think, I'm not sure if I understand the question completely. Difference of tryptophan, do you see?
I think the question is, what other different metabolites do you see? Or are you seeing different metabolites?
Yeah, I mean, within the indole pathway, I think we see the indole changes sometimes. But I mean, the other major metabolites that we discovered, we did a huge metabolomics screen a few years ago, and there's tons of different pathways that are altered, and we're just going one by one. And in the end, I think the three major ones at the moment that we identified, and they kind of account for almost like 50 to 60% of all the variation with the bile acid pathway. If that's abnormal, you for sure have a shift in the microbiome. Then we have short-chain fatty acid, obviously, which sometimes is a little bit difficult to measure in a fecal sample, obviously, but I think genetics could help us with that because we want to know which short-chain fatty producing bacteria are present. But they are often altered in some of those diseases, and the tryptophan indole pathways are still a little bit too elucidated. I mean, on the major ones, they're important, but which of the indole subtypes, we don't know yet. So we didn't have time to look at this yet, but that's probably a very important pathway.
Okay, so we have our next question. So this person is asking, we have a canine trial going on. We treat canine solid tumors with various nanomedicines. We're interested to see whether a particular treatment that worked in patient causes a specific change in the canine microbiome. Is it something that we can assess using MetaGene Canine? Emily?
So it's unclear from the question which sample matrix you would want to be testing, whether it's the tumor itself or stool. But assuming it's a stool sample or GI sample in general, I would say yes, absolutely.
Awesome. Okay, so now we have our next question is, what do you think is the impact of sampling method on the results? So I'll pass that to Emily and Jan to both try addressing that question. Emily?
Yeah, thanks, Shaima. That one is I don't want to say $1 million question, but it's a big question. And we know from a variety of studies that have been done in the human microbiome, especially, that it can have an impact. The method of sampling, whether or not you've used a stabilization method or had the ability to put your samples directly into a freezer, freeze-thaw cycles, all of these things can have impacts. And so I work very closely with colleagues at DNA Genotek who've been working on solutions to help facilitate that, especially with home or field-based collections. So it is a challenge. I think the good news is that many of these things can be overcome, but it is one of those things that can contribute to the noise in the field and the concerns around reproducibility if the impact of your collection method ultimately ends up overwhelming some of your other signals. Jan, I don't know what your thoughts are there.
Yeah, I think there's probably two components of the answer. One, maybe you, Emily, can help answering is, I guess, the amount of DNA that you need. So I think obviously for amplicon sequencing or for qPCR, we don't need much feces. We don't also need much, DNA. But I think for shotgun sequencing, you need a certain amount of DNA, correct?
Yeah, and it's gotten better than it used to be. So back when I started doing metagenomic sequencing, you needed micrograms of DNA to be able to do the work, and now we're down to nanogram levels. So
okay.
You can certainly get away with far less at this point, but you do want to make sure that you've got sufficient material to be able to support that extraction and any, if you need to go back to that sample for any reason, you want to avoid the one and done pitfall if you decide that for any reason you would want to sequence again, sequence in a different way, or go back and do some sort of other analysis to confirm your results.
Yeah, and so I can just maybe answer stability-wise. We did a lot of studies, but we focused not on the DNA. Well, I cannot the functional aspect, but I can tell you experiences which taxa are affected and so on. And what Emily said, freeze-thaw cycles are the worst. So after the third or fourth freeze-thaw cycles, a lot of DNA disappears. So it's actually better to keep it in the fridge and then freeze it really and then it stays frozen and so on. But generally, I think it's actually pretty stable. So freezing is the worst in the end, and especially repeated freeze-thaw cycles. But in the end, it's not as bad. It's actually pretty stable, reproducible in general if you keep basic principles. Room temperature is also not very good and high temperatures. So yeah, so it depends on the logistics of the trial you can do.
Okay, perfect. Thanks, Emily and Jan. So our next question is there an FMT product on the market that you trust? And if the science is not quite there, what are the next steps to have it widely adopted in clinical practice? Jan?
Yeah, that's a great question. So two answers to that. There's actually now, we're having a consortium of roughly 20 people from all over the world, from different institutions, but also private practice specialists, who do a lot of FMTs routinely. So we are combining kind of like a consortium of guidelines based on at least expert opinions that hopefully is going to be available this summer or by the fall, where we're going to recommend donor screening and procedures and which diseases do we have experiences. So for me, FMT is definitely something that is going to be used a lot. We also probably know more or less a little bit about it, meaning depends really on the underlying disease process. If it's an antibiotic-induced dysbiosis, FMT works great. If it's a chronic established GI disease, FMT can work very good, but there's often relapse rates. And again, because it's not just the microbiome that we need to change, we also need to treat these underlying conditions and so on. For me, at the moment, it depends how you screen your donor, what is a good FMT product. And again, the problem is that there are companies on the market who offer that, but the methods how they screen the donor are clearly not well established and so on. And from my experiences is that, yes, some of those products don't really have high diversity, for example, and so on. So I think for me, at the moment, the recommendation is going to be to screen your own donor when you have it with the guidelines we're going to provide. And that's going to be hopefully much easier, because then you can control really that you're really choosing the right donor based on established guidelines and not a company that may or may not follow those guidelines.
Awesome. Thanks, Jan. So we have one more question for you, Emily. In terms of what sort of data is acceptable for MetaGene Canine, can I analyze data that I've already sequenced with MetaGene Canine?
Yeah, absolutely. So if you have shotgun metagenomic data that's previously been sequenced, it can definitely be re-annotated using that solution.
Awesome. Thank you. So it looks like we don't have any more questions. I will close it off and thank you everyone for attending, and I will pass it on to Jessica.
Thank you so much, Shema, and thank you, Emily and Jan, for the great presentations, and thank you for our attendees for all of your questions throughout. If you're interested in learning more about the animal microbiome, our Animal Microbiome Connect event is happening the 21st and 22nd of June, where we'll be covering this sort of thing even further. But I hope you enjoy the rest of your week, and thank you everyone for joining us. Thanks all.